Camilla, and today I'd like to speak to you about Cryptidia fasciculata. These are protozoan parasites that solely infect mosquito species. These protozoans belong to one of the earliest forking branches of mitochondrial-containing organisms in the eukaryotic evolutionary tree. By studying these ancient protozoans, we gain a better understanding about which kinds of biological mechanisms remained conserved throughout the evolution of eukaryotes. Calcium regulation by the mitochondria can play a role in important physiological processes, such as ATP production and infectivity. The mitochondrial calcium uniporter regulates calcium levels in the cell by importing calcium ions into the matrix of the mitochondria. The MCU has been well studied in related human infective species, such as T. brucei. But by studying the MCU in Cryptidia, we may understand if there are any differences in function across biological models. Our experiments are framed based upon this question: is intracellular calcium transport regulated by a ruthenium red sensitive MCU in Cryptidia fasciculata? In order to test this, I used a calcium probe, which increases fluorescence when calcium is bound, called calcium green 5N. This only fluoresces to calcium in the cytosol. So, as calcium is transported into the matrix by the MCU, I should see a reduction in signal. Next, I used ruthenium red. This dye is an MCU inhibitor. By disrupting the MCU, I am able to see if there are any differences in calcium levels in cells with normal functioning MCU and cells with inhibited MCU. And I am able to determine the contribution of MCU to calcium uptake. These are our research goals. We expect to see a greater fluorescence signal in inhibited cells because the calcium will be aggregating in the cytosol, unable to cross into the matrix. And we expect the signal to remain low in MCU functional cells because the MCU is going to be pulling in calcium, thus decreasing available calcium in the cytosol for calcium green 5N to bind to. Primary experiments showed that all concentrations of ruthenium red were quenching the signal of calcium green 5N, making it impossible to accurately measure calcium. It was necessary to ensure that the dyes were functioning properly and accurately measuring calcium. After lowering the calcium concentration tenfold into the nanometer range, we were able to get accurate calcium readings. Samples with no ruthenium red displayed similar calcium signal as samples with ruthenium red, indicating the dye is not interfering with the output fluorescence of calcium green 5N. Once I had calibrated the optimal concentration and conditions for my reagents, I was able to work with my cells. After adding calcium in various increments, I saw that there was no difference in cytosolic calcium levels between MCU functional cells and MCU inhibited cells. We accepted the null hypothesis, meaning there was no difference in calcium regulation in cells with normal working MCU and blocked MCU. Future experimentation will be geared towards ensuring the outer membrane of the cells are properly permeabilized by a detergent called digitonin. Improper preparation may have had an effect on my results. Future research may help elucidate the precise function and role of MCU in C. fasciculata. And this is of utmost importance because we will be able to confirm the mechanism by which mitochondria regulate intracellular calcium homeostasis, which has applications in many fields of study, such as unicellular cell death pathways and bioenergetics.